We're going to open our session with Jonathan Fletcher. His bio happens to be in all the booklets, so I'm not going to repeat it. Jonathan Fletcher is a colleague, he's a friend, he's a researcher, and he's the head of the Life Raft Group research team, an incredible group of 10 of the best researchers in the world, who we brought together some years ago. We had raised a little bit of money, and the history of this team is that 4 of us, including Jonathan Fletcher, sat down in a hotel room in Totowa, New Jersey, of all places. And we had raised $2 million. And we sat down in that hotel room on a Friday night, and we turned to these 4 researchers, and we said the following to them: if you can come up with a plan, if you can agree to work together and cooperate and collaborate, we'll give you this money. And they did, and frankly it was like watching kids in a candy store, that all of a sudden researchers who have been accustomed to working to a large extent on an individual basis until they publish a paper, not only were permitted to collaborate with each other, but were mandated to collaborate with each other. We're just finishing this week the 4th year of that research team. We're about to head into our 5th year. Next week, Anita and I think I mentioned briefly last night, are going to Amsterdam as the guest of a Dutch couple. There's also going to be a cruise there, different river, and that Dutch couple has already raised so far 1 million euros that they're going to present to our research team. And we think it may be a little bit more. So the only thing we ask that you pray for in the next couple of days is the money is in euros. Please pray that the euro keeps recovering and rising. So is Jonathan here? Are you in the room, Jonathan? Oh, there he is. So let me introduce Jonathan Fletcher, the head of the Life Raft Group research team. Jonathan.
So what an honor to be present in this incredible, devoted, determined group today, and to just reflect upon a bit of what you have supported in the past 4 years. This is certainly an effort which started with a purpose and which has blossomed from there. It would be impossible to give a full sense of all of the research which is underway in these groups and through collaborations with other groups, with the intent to treat GIST better, to cure GIST, to offer possibilities for those for whom Gleevec, Sutent, and other drugs no longer work. But what I hope to do is give you a sense. All of this, one of the mandates that we discussed as a group is there's a lot that one can do to understand GIST better. The timeline here had to be critical. The work that was done, the work that was supported, had to, if all went well, help those who are in need of help 6 months, 12 months, maybe even 2 months from now, okay? And so we continue to work with that moral burden, with that sense of urgency in understanding the best ways to attack GIST. Now, up here, as Norman mentioned, there are 10 members of the team, and I'm going to guess there are 8 pictures here, okay? Because as I was scrambling at 1 o'clock this morning to find 2 more pictures. Well, I came up with one, and I'll show that in the update, a lovely picture of Sebastian Bauer, who is the 9th member of the group. And unfortunately, I don't have one of Annette, but I'm going to leave an image of Annette in your mind that will be more unforgettable, I hope, than a photographic portrait. So what were the goals then? The goals here are to develop the tools that promote international GIST research, that enable other groups, not just our own, but other groups by sharing, to do their best to conquer this disease. And I can tell you that has been a hugely successful effort. There are hundreds of laboratories who of course do not focus primarily on GIST, but have been able to make use of the tools that were developed by this effort. So as one example, it would be taking a surgical specimen and carefully managing that in the laboratory such that you can pull GIST cells out of it, put them in a plastic flask, and keep them growing so that they become an immortal resource. These are GIST cells in the laboratory, but GIST cells that came from a person and which still embody all the critical biological aspects of that person's disease, such that then you can throw drugs on those cells and you can ask, what are the best drugs that are GIST cell killers, that stop these things from growing, that stop them in their tracks? And those sorts of resources we have distributed to literally, I don't mean 10 labs, I don't mean 50 labs, to hundreds of laboratories around the world who can bring some of their scientific expertise to focus on GIST. They may work on leukemia, they may work on breast cancer, but we try to pull them in based on these models into devoting some of their terrific intellectual and infrastructure expertise to this problem that is of such concern to us. We want better therapies, of course, for children and adults who are afflicted by GIST and who are not adequately treated by Gleevec. Either they didn't respond in the first place or they responded and now have progressed. And increasingly, and this is the biggie, we want therapies that maximize the initial response to Gleevec. The sort of drug that you would add after 6 months of Gleevec treatment when all is going well to kill the remaining cells, to leverage the attack upon the GIST cells with Gleevec. We want to cure GIST. Now, to back up a moment, in Dallas, the Life Fest that I attended, I remember Norman goading us to get up on the platform in front of this incredible group and to say, we're going to cure GIST. And I was concerned. I thought, to say we're going to cure, well, first of all, if we can't cure, if we could simply do better incrementally, then we're creating false hope. And we're scientists, after all. We want to cure this disease. We want to do our best, but we also don't want to mislead with respect to the challenges that face us. We know what it means to say we're going to cure cancer. We know how hard that is. We know how difficult that has been historically for any widespread metastatic cancer. What I'm delighted to say, and in a clean conscience 4 years later, is that I do think our understanding of GIST, how these cells work, how they attack the body, has grown such in the last 4 years that now you could stand up on the stage and say with determination and conviction that we're going to cure GIST. And so just a few snapshots then of what the group has been about. As many of you know, diagnosis, although it's become much more straightforward in the past 10 years for GIST, still on occasion remains a challenge. So we still need better diagnosis for those individuals who have GIST but might be called something else to make sure that they are not deprived of the benefit of a drug like Gleevec. And one of our, oh no. This is like, I have to say the last night was a update for some of us. The mention was made about Norman tweeting, and some of us had to figure out what BFF meant. I think that was, is that correct me, James, it's best friend forever. Is that right? Yes? Okay, good. Okay. What I'm learning with the PowerPoints is that you can hide slides. You can hide slides. So my first 2 slides have been hidden from me and from you, but we're all on the learning curve with biology. I have such a cute slide of Matt Van der Rijn and his dog, his beloved dog, which was the rationale for naming an important gene that his group discovered in GIST, that just like KIT is a very specific marker of GIST. It's called DOG1. We can see it in this slide here. But this gene coming out of the efforts here has rapidly entered the diagnostic lexicon for GIST. So if you have a tumor that in the eyes of a specialized pathologist looks like a GIST, but doesn't stain with KIT, that's the classic marker that makes you comfortable with the diagnosis. Now you can use this new, straightforward diagnostic stain for a protein called DOG1 that puts you on very sure ground. And this large pie type of diagram at the lower right is showing us that out of 1,546 GISTs that were reviewed, 85% of them, that's the big black area, stained for KIT, the classic marker. And that left us with a subset, no, actually 88%. That left us with 12% of the GISTs overall that did not have the KIT protein and therefore are not so straightforward to diagnose. Half of those, or 6% of the total, have very strong positivity, strong staining for this new marker, DOG1, which again is a very specific marker for GIST. So we're increasingly whittling away at that subset of GISTs which are not straightforward to diagnose, to make sure that everybody gets the right therapeutic options. This is also a chance to mention over on the left, you see Matt's lab group as of 3 years ago, and one of the wonderful aspects of this democratic approach and interactive approach to research is that we even exchange some of our most capable laboratory members. So the guy at the far left with the yellow arrow on him, who is just a fantastic GIST scientist, Cheng-Han Lee, started in Matt Vanderrijn's lab at Stanford, and he's now been in my laboratory for the past 2 years. We swapped. And now he's going to move shortly to set up his own GIST laboratory in Vancouver, where his family is from. How do GISTs develop? Now, this is perhaps the one area that you might think has the least short-term relevance to progress in GIST. If the goal is we want better treatments for those who need them today and we want them next month, then to understand what kind of a cell does a GIST come from may be a longer-term sort of effort, and I think that's true, but it's an incredibly important one, because as we find new ways to attack GIST cells, in the GIST tumor, it's only a small population of the cells, a small percentage of the total, that are actually the starting problem. And then they are giving rise to other cell types within the tumor. But that early population, and increasingly in biology, those are called stem cells, the stem that gives rise to all the rest. If you have a new therapy that attacks the GIST cells in some different way, you sure want to know that you're attacking the start of the problem, those stem cells, because if you can take those out, then you destroy everything else. You know, it's like a house of cards. If you pull out the bottom, then everything else falls down. So in the longer run, it's remarkably important to understand what are the origin cells within the tumor that are multiplying and giving rise to all the rest, because those are the ones that we want to make sure are well covered. This is a good moment to mention the incredible importance of an entity called microGIST for our research, and I think strategically for pushing GIST forward. GIST, of course, is a clinical problem we all know is a rare disease, and so it's very helpful to be able to go to the big funding organizations, the National Institute of Health, that release dollars to our laboratories beyond those that you have all supported, and to be able to say, yeah, it's a rare disease, but beyond having an incredible target with KIT and PDGFRA, which we don't have in most cancer types, beyond having a dream drug in Gleevec, to be able to shut that target down, at least for a good long while, for most, not all, but for most, there are other things that make GIST an incredible, a remarkable model in cancer. So please give us more money. You know, you have to make those arguments, and you have to convince people when you have a rare disease model. MicroGIST, this is one of those arguments. Do you know that 30% of the population, I'm not talking about in this room, I just mean walking on the streets over in Manhattan or Jersey City, 30% of the population have a KIT mutant GIST. It's a small GIST, but biologically, it's so much like the clinical problem that many of you in this room have had to deal with. It's the same cells, the same kind of mutation. The thing that doesn't happen for the 30% of the population, of course, we know this statistically since it's a rare disease, the thing that doesn't happen is that these small little true GISTs, 3 millimeters in size, 1 centimeter in size, they don't generally progress to become a clinical problem. What's the frequency of GIST in the general population? Real low. What's the frequency of microGIST? This tells us that you can start with a KIT mutation, but then there are some other critical pathways that need to be activated in those cells to push that starting point along to becoming a clinical threat. And so by understanding what those pathways are, of course, it gives us more options for points of attack. And then real important, by having this model that we can work with of the GIST at its earliest point, and by knowing what cells GISTs arise from originally, we can, as we find new problems in the big bad GISTs, in the clinical GISTs, we can ask, are those problems already present in the earliest stages? If they are, we're more enthusiastic, because that sounds like it's of core importance to the GIST. If it's a problem that only develops late in the process, then that starts to sound, even though it might be useful as a point of therapeutic attack, as though perhaps it's less central to the development of GIST and therefore may do less harm to the cells when we attack it. Okay, I mentioned the stem cell, and I'm not going to belabor this, but gorgeous work from Brian Rubin's group, one of the members of the team, using mouse models that he's developed that have KIT mutations and that develop GIST to identify exactly what are the precursor cells, what are the stem cells that we most want to go after in GIST. I'll just say a few words, and it's really very much a partial snapshot about wild-type GIST. One thing that's become so much more clear in the past 2 years is that by studying pediatric GIST, we learn a lot about adult wild-type GIST and vice versa. You know, it's no longer the notion that you need to work on pediatric GIST even though it's ultra rare because Gleevec doesn't work as well and Sutent doesn't work as well in that population. Now, even from the adult GIST side, we can say that on both sides of the divide, pediatric and adult, if you want to do your best, you have to understand these two groups. You have to understand them essentially as one group, again, the pediatric and the adult wild type. And just to mention one area in which Christina Antonescu, who's at the Memorial Sloan Kettering across the river here in Manhattan, has made some beautiful progress. Christina has showed that a gene which is, you know, we can think of it as being downstream of KIT. KIT is a protein which is at the surface of the cell, and part of the protein pokes out of the cell. And when KIT is turned on, when it's activated, like by the typical mutations in GIST, then it sets into action a series of events that move energy through the cell and ultimately tell the cell to grow. You know, the things that we don't want the GIST cell to do. And BRAF is one of those things within the cell that KIT passes its message to, and then BRAF passes that energy on to additional proteins within the cell to tell them to grow. Christina has shown that in a subset of the wild-type GISTs, those that don't have KIT mutations and don't have PDGFRA mutations, that this BRAF instead is directly activated by mutation, about 7% of the total group of wild types. Okay. And then George Dimitri mentioned yesterday the spectacular example, one of the few outside of the GIST arena, where in melanoma, in a lethal form of skin cancer, there is a drug now which has Gleevec-like qualities, the sort of thing that Gleevec does for GIST. One can see it in this melanoma. Do you know what that drug is attacking? It's BRAF. There's a spectacular drug now for BRAF. So this makes us very enthusiastic that even though we're talking about only a subset of the adult wild-type GISTs, here is a situation that's primed for a new therapeutic approach in GIST. The problem of resistance heterogeneity, and this was really what caused this working group, this research group, to come together. The realization that when an individual has had a nice response to Gleevec, and then 2 years later perhaps might have a progression, that the resistance mechanism isn't one thing. It's often several things. So it may be a new mutation in KIT in one area of the progressing GIST, and a different new mutation in KIT in a second area of the progressing GIST. There are a number of different events that can contribute to this resistance problem. And that gives rise for a need for new types of therapies, because in addition to drugs that work like Gleevec, nilotinib, sorafenib, and so forth, we need drugs in the face of altered forms of KIT that attack the protein fundamentally in a different way. And that's why we're delighted, in addition to the remarkable support that Novartis has provided and Pfizer has provided over the years for all of this work, that there are companies who have a newer stake in GIST who have been able, you know, sort of taking, you know, suspending doubt in a way, because this is a rare disease model, but being very convinced by the opportunity, the need, and the biology, have been willing to enter the fray. And Dan Flynn, who is the CEO from Deciphera, a company which is developing a remarkable new group of drugs that attack KIT in different ways, is attending the conference today, which we're tremendously grateful for. So the drugs like Gleevec and Sutent, they bind to a part of the KIT protein which is called the ATP binding pocket, and Deciphera has developed a number of drugs which bind to a different part of the protein called the switch pocket region. And the attributes of these switch pocket inhibitors include that when they bind to KIT, they're pretty much stuck on the protein. Certain drugs that inhibit KIT, they bind, and then they come off reasonably quickly. These ones are more like glue, okay? So they do the job extremely effectively. And a protein like KIT exists in what we refer to as an activated state and an inactivated state. Some drugs bind only to one state or the other. The switch pocket compounds, as far as we can tell, bind to both of the states. So one has a sense of a kind of super KIT inhibitor with these new forms of compounds. And in studies that Mike Heinrich, one of the members of the research team, just presented at the lead clinical oncology meeting, the American Society of Clinical Oncology, the following results were demonstrated. These switch pocket inhibitors, most remarkably, inhibited every single Gleevec and Sutent resistance mutation that we have yet identified. Now, I'm not talking about in patients. We're talking about laboratory studies. But there is no other direct inhibitor of KIT that we've come across that shuts down all of these resistance mutations. So this is remarkably encouraging. The preliminary pharmacology and toxicology studies with these drugs have been encouraging, which means that when you administer them in animals, they act like drugs. It seems like they would work in people. And in vivo, that means in animals, efficacy studies using GIST xenografts and other GIST models are now in progress with the intention that by the later part of this year, we want to have identified the best drug in this incredibly promising series to push into a new clinical trial. So, let's keep our fingers crossed, but I think this is an approach, is one example that has remarkable promise. If all works well, certainly if these drugs work as they do in the laboratory setting, then we have a very, very powerful new member of the GIST armamentarium. Okay? Annette Dunsing. So I mentioned that I would try to leave an impression that was stronger than a photograph. One of the wonderful things with the research support has been that we have been able to encourage collectively new, determined, wonderful scientists to set up their own GIST labs that work only on GIST as a problem. Understanding that cell, new therapies. They've got 10 people in their lab that work night and day on the GIST problem. So when you think about it, the support for GIST research, which is seed money, which enables new groups to do their thing, is just a remarkable opportunity. Annette's one of those. Annette is from Germany. And the impression I'll leave you, when she first came to see me, it was 8 years ago, looking for a postdoctoral fellowship in my laboratory. Annette disagrees with this, but I'll swear to you, she had a black leather jacket, black leather pants, and very, very East German at the time. And I thought, my gosh, this doesn't look like a budding scientist. She looks a bit wild, in fact. Oops. So I had my doubts. Well, Annette Dunsing, for any who have met her, who've had that pleasure, she's since graduated from my lab, did seminal work on GIST biology in my lab. She has set up her own full-time laboratory at University of Pittsburgh. She not only works on GIST, but her brilliant husband, Stefan, they run this laboratory together, has been pulled for his work, which was on how cells grow, into the GIST field as well, and that has yanked him into the GIST field. And she's about the kindest, most thoughtful, considered person that one could imagine. But you will meet Annette for yourself, I hope. What Annette has done in the past 2 to 3 years is to emphasize, what about those cells when you've had a response to Gleevec for inoperable GIST that remain? Those cells that are actually drug sensitive because they're not growing anymore, but they persist. They aren't dying. What do we do to kill those cells? And she calls that state quiescence, where the cells are alive, they're still there, they're not growing, but neither are they dying. And that has blazed the way in this all-important challenge that we have of adding a second drug to Gleevec to kill off those remaining cells. One of the nice, and I know in the Monterey meeting, Norman and others were thrilled by this slide, because one of the nice approaches that Annette has developed, very powerful, is to show GIST cells in culture. Remember, I mentioned before that we have established GIST cell lines from patient materials, and she follows them over 72 hours by time lapse, so that when you add a drug, you can determine just visually, are the cells going to hell in a handbasket? And in this series, we're going to see GIST cells that start, in our eyes, looking robust, and at the very end, they kind of widen out and become circular. They're like fried eggs, and those are very sorry, sad GIST cells. Why? Because Annette has added Gleevec to them. So this platform then enables her and others to add other drugs and again to track, are they hurting those cells in a very short time period? You can determine that. So let's watch. Okay. This is probably the first day. I wish I had a pointer. And now we're coming into perhaps the second day. These cells are moving around a little bit in the plastic tissue culture dish. There it goes, okay? You see that? This, did you see that widen out? Okay. So after about 2 or 3 days of treatment, we had GIST cells that before had been robust, they'd been hardy, and now they're widening out and they're stuck. They can't grow anymore. Okay? So this is a terrific thing to see with a drug, and as we're screening new drugs, then we want to see exactly that, including for GIST cells that no longer respond to Gleevec. Of course, ultimately we want the drug where after 3 days there's nothing left on the plate, right? You know, the cells are totally toast, but we'll also settle for stopping the cells entirely in their tracks. Okay? HSP90, we're near the end, so you know, and I should have said at the outset, I thank all you brave souls for getting up early and listening to a research presentation, since a new field in science isn't always easy for any of us. I appreciate that. Here's one which is perhaps a bit tough conceptually, but it's real important, I think, to have a sense that HSP90 inhibition has huge promise for the GIST field. We know, some of you, that the initial trial of IPI 504 was promising, but not a home run. There is a new trial of STA 9090 sponsored by Cinta, which we're hugely grateful for, because different ways of inhibiting the HSP90 have their own advantages. And one of the things that Cinta has supported and encouraged, which we need so critically in this field, is to offer, through George Dimitri and his collaborators, optional biopsies done right before the first drug is started and then after the second dose. We need to know in HSP90 inhibition why these cells aren't being blown out of the water so that we can move the field along, and we need patient material to determine that. So we have that in hand now, and I think it's going to drive the field forward hugely. But what do HSP90 or heat shock proteins do to KIT? They basically hold the hand of KIT in the cell and make sure that it functions properly, that the protein itself is in the right structure, it's in the right place in the cell. You know, things that are turned on by mutations in cells, the cells try to destroy them, and HSP90 makes sure that the cell doesn't do that. So it's the protector of KIT. That means that if you hit HSP90 hard with a drug, and it certainly works in the laboratory, you destroy KIT in the GIST cells, and you hurt the cells badly. We want that to happen, of course, now in our patients in clinical trials. Okay, and just to emphasize this point, on the left we have HSP90, two proteins bound to each other as they're found in normal cells in our body. And over on the right, we have HSP90 in the supercomplex. All those other circles are other proteins that in the GIST cell bind to HSP90 and make it extra effective as a watchdog for KIT. So it's not just that you can attack HSP90, you could potentially attack some of those other proteins that accelerate the function of HSP90 as a way of derailing KIT. And we need to learn a lot more about HSP90 in a hurry to be able to figure out the best point of attack. Okay? So Sebastian Bauer, now we have a photo of him. One thing, Sebastian Bauer also spent some time in my laboratory. He's the 10th member of the research team. He now has his own full-time, very productive laboratory in Essen, Germany. And I can tell you, you can have full confidence that this guy in his lab will make important progress in GIST. When he came to my lab, he had 3 kids in 2.5 years. Not he, his wife had the children, but they managed to do this while both being productive in Boston. And this is, with Sebastian, is Paul, his oldest, and Mary Lou, his middle child. Bela, his youngest, is not in that picture. Bela's now 5 years old and goes on 25-mile bike rides, you know, not in tandem. I mean, he's on his own bike. So this is an energetic, vigorous family, and Sebastian has done already heroic work, I would say, since setting up his own GIST laboratory. What he has shown is that another form of attack, and this is real important because a clinical trial is about to open if it hasn't already, called HDAC inhibition, has a lot in common with HSP90 inhibition. And for all we know, it might turn out to be a better way actually to inhibit HSP90, to do some of the things that we've wanted with those drugs. HDAC, it's a mouthful, it means histone deacetylase. Histones are on our chromosomes, and HDAC would normally remove acetylation. If you inhibit that process, which we're doing with the drugs, then you have too much acetylation, which is an add-on to the proteins and the histones. And what Sebastian has shown is that even though these drugs are thought to target histones, they also target HSP90. They affect acetylation of HSP90, leading to increased acetylation, and an acetylated HSP90 is essentially a broken HSP90. It can no longer do that job in shepherding KIT along. So he's shown that when you use an HDAC inhibitor in the GIST cells, you blow KIT out of the water. Of course, the cells are unhappy then. And we're really excited that Novartis has arranged, under Sebastian and others' direction, a trial of their terrific HDAC inhibitor, LBH589, which is the first in GIST. And Jenna, you may know whether that's open now or whether it's imminent, but we understand that this is, if it's not open, it's just about to open. Okay. The last subject that I'll touch upon to give a better sense, we've touched on some topics, some promising targets in the GIST cells, but just a slightly better sense of the complexity of the process and why it helps to have a team working together, as Norman has emphasized. We don't just analyze the GIST cells in one way. We work with the cells growing in culture, with surgical specimens that come from patients. We analyze the genes in those cells, the proteins in those cells, and increasingly, those give rise, with so-called high-throughput approaches, to complex data sets. And you need strong cooperation between groups, not to mention bioinformatics, to tie all those data sets together and to identify the best opportunities. What are the best opportunities? They tend to be the things that, no matter how you look at the GIST cell, they come up as an important factor. So you might look at the protein level, at the gene level, at the growing cells, at the cells in the patient. Any way you look at it, you're going to find out that KIT's a biggie in the GIST cells, of course, and we want more core biological processes in the GIST cells like that. So we're using a variety of those approaches, and this is the last, I think, fairly tough topic that I'm going to introduce, but again, one that I think, looking for the next 2 to 3 years, is extremely important. The topic of synthetic lethality. Synthetic lethality means that we have created a state, we have synthesized a state where an event, an attack upon the cell, becomes a lethal situation, becomes a killer. Okay, so in the ordinary situation, that attack upon the GIST cell may not phase it, but you set the cells up in some way. You change their biology such they're like primed for this new drug. And in that special situation, that special context, it does become a killer. Synthetic lethality. So one of the things we do to accomplish this is that we ask the question, by treating our GIST cells in the laboratory with a rather moderate amount of Gleevec, what are those processes in which the GIST cell remains alive? And then we treat with a second drug, and that's the question mark down here at a dose, again, where the GIST cells would still be alive. But we look for situations whereby combining them together, combining the two drugs, and again, the second drug, importantly, would not hurt the GIST cells on its own, would not hurt normal cells on the body to any extent on its own, but as shown in the bottom row, combined with Gleevec, with a modest dose of Gleevec, in that special situation, the synergy between the two, that becomes a killer package, okay? Obviously, as we're looking for new useful therapies, since we have no guarantee of being able to cure GIST for most within a certain time frame, at the very least, we want more individuals who can live with GIST and feel well for 20 years, for 30 years, and that means very selective drug combinations that are well tolerated. You have to be creative and clever about this. Find a drug that, again, on its own wouldn't do much, which means perhaps not much toxicity, but combine it with something else that's well tolerated, like Gleevec or nilotinib to Cigna, and which on its own may no longer be doing the job fully, and where the package, which is still well tolerated, is enough to do the job together. So in one approach, we are actually knocking down or eliminating 11,000 different genes in the GIST cells, a huge number of genes. And for each gene, we're doing that 5 different ways to ask the question, what are the critical genes beyond KIT and PDGFRA, IGF-1R, in the GIST cells? And then we're asking the same question after we treat the cells with a dose of Gleevec, which again is enough to affect them, enough to change their biology, but not enough to outright kill them. And we're asking, now what are the genes that in the untreated cell, when we eliminated them, did nothing to the GIST, but once we have primed the cells with a moderate amount of Gleevec treatment, become absolute cell killers. Much to our surprise, when we did the first study of 11,000 genes and the first of our GIST models, and we need to do substantially more of this, I might add, we found that out of 11,000 genes in the cell that were tested, the one that was most essential to GIST, so if you knock that one out, the GIST cells went absolutely belly up, was a protein called CDC37. CDC37 was one of those bubbles that I showed around HSP90 in the GIST cells, one of those things that accelerates the function of HSP90. And the remarkable thing here is that CDC37, although HSP90 itself, I've emphasized it being a shepherd for KIT in the cells, but it's also a watchdog for about 100 other proteins in the cell, depending on the cell type, which again suggests the possibility, which we know, of toxicity with some of these compounds. CDC37 is the thing that takes HSP90 and says, only worry about the kinases, the proteins that are like KIT, a subset of what HSP90 usually looks after. So this provides CDC37 as a point of attack, more specificity as a concept, we think, than going after HSP90 itself. Okay? And here in this study, we have knocked out CDC37, this new potential target, by several different approaches. At top, we have untreated GIST cells, and that's a rug of cells after 10 days of control treatment. And at the bottom, we have eliminated CDC37 by 2 different approaches. And you can see that the GIST cells here have nearly disappeared. They're very light on the tissue culture flask compared to the untreated cell. Is there anything we can do now as an attack upon CDC37? Unfortunately, the answer is no. There is a Chinese folk medicine called the Thunder of God vine, which is used primarily for inflammation, a pretty toxic folk medicine. And one of the components of this vine is celestrol, and celestrol is a CDC37 inhibitor. We've tested it in the lab, and it works beautifully to destroy KIT and to destroy GIST cells. But our sense is that in addition to inhibiting CDC37, the thing we want here, it also does other things to the cells. It's not selective enough. So as a first step, we don't think this is going to be useful clinically, but we sure as heck want to find selective drugs that derail the interaction between CDC37 and HSP90. Okay. In my last 3 slides, and Norman, with your blessing, okay, we were in Vienna. When was it? I can't keep track, but I think last weekend. Yes. Okay. And some of you in the room were as well. Many of you in the room. Some of you. And the question that Norman put to a number of us with this remarkable influx of research support, the at least €1 million, which, although of course it doesn't allow you to do everything, remarkably increases the possibilities. How do you use that best to serve the needs of this room and to drive research along, not just in this research team, but for other teams who are pushing the GIST possibilities as well? And in part, as the Life Raft Group board and Norman have envisioned it, this amounts to providing funding from this bucket to groups who are outside of the research team, to expanding the number of groups who can be supported. But critically, it involves seeking opinions from experts at every level, scientifically and medically, to weigh in where do we get the most value, particularly if we're thinking about a payoff in 6 months, in 12 months, in 18 months, not something that is helpful 10 years down the road, which of course is nice, but may not be the first priority for some of the folks in the room who would rather see a better option 2 months from now or 3 months from now. And so here are 3 possibilities that we are convinced would help. And just to illustrate to you that this is not a straightforward process, I want to cover them quickly and mention for each why there are huge opportunities, but why there are some challenges. This tends to be the way in science. So one of the approaches is simply to take our wonderful GIST models, which include the cell lines and GIST in mice, and screen thousands of different drugs. You can do this with tens of thousands of drugs, actually, using so-called high-throughput platforms, to look for drugs that are already out there, and preferably those drugs that are already FDA approved, that can be moved rapidly into a new indication, to see what there is that we don't know of that is a GIST killer. I mean, that sounds like a fairly straightforward process. And personally, I hope that some of that work will be supported with these research monies, because that provides the sense of perhaps the most rapid payoff. The problem with these studies is that as you screen tens of thousands of compounds, you might not be surprised to know that many of those aren't drugs actually. They are compounds you can use in a laboratory, but they're not drugs from the standpoint of a person. They won't work pharmacologically, and in many cases they may kill the gist cell, but we'd have no idea why. That might not seem important, but it ends up being really important, and it's part of why Gleevec has been such a success, to know what you're attacking and to be able to use that wisely and build out from there. Okay, the gist genome. So sequencing the genome, every piece of the DNA, of course, is a current topic of interest in almost any cancer. We're learning that as the genome, all of the DNA is analyzed in certain types of cancer, like typical lung cancers, the complexity of mutations is unbelievable. And in a certain respect, that might cause you to say, well, we can sequence the genome, sure, it's a bit of an effort, but will there be so much going on that we won't be able to chart a course? We won't have a sense of direction from that data. And almost. And we think maybe, but GIST has proved, as we know, to be a simpler model than most cancers. It's just as deadly. It's just as much of a problem. But the genome has been simpler. And our sense that by sequencing the genome, we may find new kits, things that have mutations and things that are druggable. Our sense of that is that this is a good possibility. Okay, and the last one, I sketched this out in the last few slides, so I won't go into in depth, but in a way you can ask, what about the functions of genes in the GIST cell? Not just whether or not they're mutations, but each gene, is it important to the cell or is it not important? Are there some genes like the CDC37 which appear to be incredibly important to the GIST cell and which right here and now are druggable? We already have drugs. If we just knew what gene it was, we would be able to pick the drug that's already in a pipeline and apply it in a clinical trial. Of course, much of what's critically important to the GIST cell we won't have a drug for. So as we do additional screens of this nature, much of this will be the underpinnings of successes that will be years off, but we would hope that some of it would translate into new therapeutic opportunities just in the next year or so. Okay? So with that, now I'm done. And I'd like to thank my own lab group, all of this is just a portion of those who work night and day on the GIST problem in my laboratory. I'd like to thank certainly all of the companies who have contributed to this work, who have provided their own support, which, you know, again, at the end of the day, it's a rare disease, and we have to be profoundly grateful to the vision of companies like Novartis, Pfizer, Cinta, Decipher, and others, that they are willing to commit to something that, in terms of a market equation, may be less straightforward than going after a target in a much more common form of cancer. But they're convinced by the need, by the way all of you have coalesced to support this, which creates a bigger opportunity, actually, a sense that if you have something which is good, it will be used and it will be appreciated, certainly. So we're profoundly grateful to that, and of course profoundly grateful to the Life Raft Group and all of you for support. Now there's a reason for this. George Dimitri, yes, this is my segue to the talk that you really would like to hear, which is George Dimitri's clinical talk. And you don't get the Humanitarian of the Decade award unless you're a very, very busy people. Note that, you know, two of probably the busiest people on the planet, Vasila and Dimitri, got the Humanitarian. So you don't get that by sitting on the couch. It's a rare occasion when you can catch George Dimitri actually relaxing. But I will point out that this was after a morning of giving teaching seminars on GIST, on the Dead Sea in Jordan, and there was an afternoon to follow of giving teaching seminars in GIST to bring docs from Iraq, Saudi Arabia, Jordan, Lebanon, and other countries up to speed on the current ways to treat GIST best. And George only had about 20 minutes here, so this wasn't a full day of relaxation. Okay, thank you.